Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Luana Maroja. She is professor of biology at Williams College. She is interested in the evolution of barriers to gene exchange, speciation, population genetics, and phylogeography. Her study systems are crickets, artic plants, and butterflies. And today we're going to talk about the topics I just mentioned. So, Dr. Maroja, welcome to the show. It's a pleasure to have you on.
Hello. Thank you so much for inviting me, Ricardo. Okay. Do we have questions?
I'm going to start now with the questions. So let's start with gene exchange then. I mean, what is it? When does it occur? Tell us about it.
So gene exchange is simply the flow of genes from one population, or in my case, sometimes I study gene flow between species, but it's just defined as the gene flow. So the flux of alleles or different kinds of genes that do not exist in one population to another. So people think of it as migration, right? I'm a migrant. I come from South America to the US. I reproduced in the US. Therefore, some of my genes that include genes that are more unique to South America, like indigenous American, are now in the US. So there was like a flow of genes from one population into the other. It doesn't always have to be caused by the movement of individuals. So you can have gene flow because you have a continuous group of individuals through space, and genes from one end of the population flow to the other end through continuous mating in space. So it doesn't necessarily involve migrants, but it's easy to understand if you have migrants involved, right?
Yeah, but I mean, apart from reproduction, what are some of the other common forms of gene exchange that we find in evolution?
So obviously you can have gene exchange caused by lateral gene transfers, which are some rare events in which genes from one species can get transferred to the other. My favorite example is with aphids. So aphids, those little bugs that are pests in garden, they actually have a gene that comes directly from fungus. So obviously there was no mating between a fungus and an aphid. These genes allow them to manufacture keratins, which these insects cannot do, but fungus can. So with that gene, they're able to survive on a diet that has zero keratins, because the phloem they eat has no keratins at all, and yet manufacture their own keratins producing colors like green, yellow, red, and that's exactly due to this lateral gene transfer from a fungus into an insect. So there is one form of gene flow that does not involve mating whatsoever.
But what are the conditions where gene exchange can occur between different species?
So when different species are closely related, they can still produce hybrids. So I think everyone is familiar with the Neanderthal human mating. Neanderthals and humans were different species. They were able to mate, and at least some of the offspring, there is some evidence that the male offspring did not fare well, which is a common pattern in mammals, that when you have hybrids, the female hybrids tend to do better than the male hybrids. So Neanderthals and humans, different species, morphologically very different, with separate evolutionary histories, were able to produce hybrids. And in fact, all humans out of Africa have about 2% Neanderthal DNA. So that was a successful hybridization that we still carry gene flow from Neanderthals to humans present in all humans around the world.
Mm-hmm. Can gene exchange also occur through infection, for example? Because I've heard, I don't know if this is true or not, but I've heard that a certain small percentage of our DNA comes from viruses, for example.
Right, so that would be another example of lateral gene transfer. It's very common. So we carry a number of genes that were once genes of parasites, viruses in particular, that are now part of our own genomic DNA. Many of them don't do anything. Occasionally, there is an advantage to some of these lateral gene transfer. Most of them are just, they don't do good or bad. They're just there.
Mm-hmm. But I mean, in this case, for these genes to become part of the human genetic pool, let's say, they have to infect germline cells, right?
Right, right. So the infection must have happened in the ovaries of someone or the testicles of someone in order to end up in the gametes, yeah. Or perhaps sometimes during early embryonic growth, right? So if a very recent embryo is infected and acquires that, then the whole body of that individual is going to contain those genes because it happened in a few cell stage. So that's another way.
But I mean, when is it that when we have these lateral gene exchanges, I mean, the genes stick to a particular species gene pool? I mean, because couldn't it be that some of them are just nefarious or have neutral or are neutral and then eventually they get wiped from the gene pool? I mean, when do they stick, basically?
So they will stick either because they have some kind of an advantage, you know, like allow a new kind of protein to be made, something like that, or because, like the case of aphids, for example, was a huge advantage to get the keratin gene from a fungus, or because they don't cause any harm, so they can stay there. So in the case of the Neanderthals and humans, we have studied that very thoroughly. And it's a very interesting story, right? Because many genes were eliminated. So there is evidence of very strong selection against Neanderthal genes, again, pointing to the fact that we were indeed two different species. It wasn't all harmonious. The hybrid wasn't great. The ones that remained, the Neanderthal genes that remained, some of them were under positive selection. So for example, genes related to skin, metabolic functions, and hair texture, actually many of them come from Neanderthals. And it might have helped humans adapt to the new environment in which they were migrating into, but Neanderthals had been there for a long time. Whereas other genes were genes in Neanderthal that were evolved for their genomic background and did not work well on the human genomic background and were therefore eliminated. If you look at our X chromosome, it almost doesn't have Neanderthal genes, which is, again, pointing to the fact that perhaps males didn't reproduce as much, limiting even more the number of, and there are no Y chromosomes from Neanderthal either.
So it's basically the same kind of natural selection under which the other normal, I mean, genes get selected, right? Because I mean, for example, if it's a case of infection or something like that, an individual or several individuals might get infected, and then those genes get transferred to their germline cells. And then if it happens that those genes confer their descendants some advantage in the kinds of environments they are exposed to, then they stick to the gene pool of that species or population, correct?
Or if you happen to have, for example, a chance occurrence. So again, the Neanderthals, humans, right? As the humans left Africa very early on, probably somewhere in the Middle East, they encountered Neanderthals, they mated, there were few humans, and now that one hybrid was actually a substantial proportion of the human population. It survived, mated to other humans, and eventually all humans were carrying that Neanderthal DNA, right? With the infection and lateral gene transfer, you could have a similar kind of thing, like you had a small population that was then expanding. One of the early individuals had that lateral gene transfer. As long as it's not too detrimental or not detrimental at all, then a lot of the descendants, because the population is expanding, could inherit that. So there is another way in which genetic drift wouldn't eliminate that allele, even if it was detrimental, if the population was, even if it was slightly detrimental. Too detrimental selection would eliminate. But if it was neutral, then an expanding population would have low chances of losing the allele, right?
Yeah, and particularly when it comes to these interspecies gene transfers, I mean, it's a little bit coincidental that some of these genes just happen to produce proteins that then give rise to certain phenotypes that are advantageous, right? I mean, it's just coincidental.
Right.
Yeah, because I mean, even the same gene, I'm not sure if this is right or not, but even the same gene in different species might not produce the same amino acids or proteins.
Right. So if the species are closely related, chances are the genes per se are the same, but the alleles are different, right? So when we talk about the Neanderthal genes, it's not a gene that humans don't have. It's a variant of the gene that humans don't have. So a different allele. So when we talk about, I don't know, like hair texture genes from Neanderthal, it doesn't mean that humans didn't have it before. It's like it just have a different version of that particular gene.
Yeah, and perhaps many of these genes, I mean, sometimes I hear people talking about the junk DNA. I mean, perhaps we could get into that conversation because it's arguable if it's really junk DNA or not, right? But perhaps some of those genes just so happen to stick to the gene pool of a particular species and they don't participate in anything really relevant, but they're just there. Right.
Right.
Because they're also not nefarious or don't produce anything that have some sort of negative consequences for the individuals. Yeah. So, and what about barriers to gene exchange? I mean, what are some of the main ones?
So barriers to gene exchange is between species, right? So you can have barriers that are extrinsic to the organism. For example, mountain ranges or rivers or oceans, you know, that there cannot be gene flow because there is an extrinsic barriers. The kinds of barriers that are studied are intrinsic barriers. So there is barriers that prevent individuals from different species from mating with each other. And traditionally, these barriers were seen as post-zygotic barriers or pre-zygotic barriers. The pre-zygotic ones happen before the zygote is formed, so before mating. And there's a ton of them. So if you think of behavior, for example, I think most humans would not consider dating a chimp because behaviorally we're so different that there is no way that, you know, that this would happen, right? So there will be a behavioral barrier. In animals, you'll find that different species have different songs, courtship songs or behaviors in fruit flies. I also studied some fruit flies that have different wing frequency when they are doing their courtship, and females of one species find that completely not attractive. And it's really funny because if you actually clip the wings off and you play the song of the other species, then they mate right away. There's no problem at all. It's strictly the song. So you have that kind of barrier. You have barriers because of temporal isolation. So for example, two different species of very closely related flowers may flower in different parts of the year. So even though they could mate, they never have flowers in the same time. So that's a temporal isolation. If you think of ocean creatures, there is a lot of that. Some of them will release the gametes in the early hours of the morning. Some of them will release the gametes in the late time of the morning, and those gametes never meet each other. So that's another possibility. If you do it in the lab, they can actually create hybrids, but in nature it never happens because of these time differences. Other barriers include post-zygotic barriers, and I think everyone is familiar with the hybrid unfitness, right? You think of the mule, ligers. There are many kinds of horses and zebras. There is horses. There is donkeys, horses and donkeys. So all of those have sterility. Most of the individual hybrids are sterile. And of course, that's a barrier. Even though the hybrid is produced, it's fine. It doesn't mate back. So there is really no gene flow. While there is a mating and a hybrid created, that hybrid does not contribute to gene flow between species. So there can only be gene flow between species if the hybrids are at least partially hybrid, partially fertile, and can mate back to some of the species. So in the crickets that I work, there are two species. There's the beach cricket and the field crickets, and hybrids can only be produced from field crickets. So the beach crickets, if they mate with the other species, the eggs don't hatch. There is no fertilization. But one way produces hybrids which are fit, and they tend to mate back with one of the species. So there is gene flow between the species in one direction, basically. So that would be, so that would be an example of hybrids which are at least partially fertile and can contribute to gene flow. Other kinds of barriers include an interesting kind that is post-mating, but pre-zygotic. So a female will mate with the other male, with the male of the other species, but then inside the female, there are things like sperm competition. So a lot of people don't know that most organisms, pretty much all insects, can store sperm for life. So the female can mate once and carry the sperm of that male for her entire life. And they have organs that can actually hold the sperm and keep it viable. And inside those structures, you can have sperm competition where the sperm of the same species does better than the other species. So if the female mates indiscriminately, including both males of her own species and the other species, most of her offspring are actually going to be of the same species. So that's another kind of barrier. Conspecific sperm precedence is called. It also happens in plants with pollen. So some plants, they will produce hybrid offspring just fine if they receive just pollen from the other species. But if they receive both pollens, then the pollen of their own species win the fertilization race, and the seed is not hybrid. So there are all kinds of barriers that are very interesting, and there's huge variation between creatures.
Yeah, about what you said, two questions. So when it comes to the hybrids, are some of the hybrids you mentioned between some of the species you mentioned, are any of them, or can they become fertile or not?
Yeah, so some hybrids are fertile right away. So for example, my crickets, the hybrids that are produced are fertile right away. The females are fully fertile. There is no problem, at least in captivity, right? In the field, it's harder to study if there is a difference, but in captivity, we do not observe any difference at all. So definitely some hybrids can just be fertile. In fact, like even the case of the mule, about 1% of the mules are actually fertile. I don't think that we understand why, but occasionally it happens that one is fertile, but it's a very small percentage. In some species, and in other species, all of them are fertile. So,
but is that enough for us to consider it a new species, or is it even a new species or not?
Yeah, so species definition is a whole other complicated thing because of these cases. So speciation is a continuum, right? So there is like a level, a giraffe and an elephant, that nobody questions that they are different species. Then you get different populations of giraffe. Are they different species or not? They have different spots. They're morphologically different. Are they different species or not? So you have the biological species concept, which says that if you can produce fertile offspring, then you are the same species. So if there is no barriers at all and you can produce fertile offspring, then you are the same species, period. The problem is that, like in the case of my crickets, right, you can produce fertile offspring, but only in one direction. Only if the females are field crickets and the males are beach crickets, then the offspring are fine. But in the other way, there's no offspring. They're actually not viable at all. So in this case, I would say that they are different species, even though one of the barriers is porous, is permeable. So there are many situations in which the barriers are not really barriers like a wall. They are permeable barriers. They block some, but not all of the gene flow. And where do you set the rule? This is a species, this is not, varies a lot depending on the researcher and on this favorite species concept. There are dozens of species concepts. So depending which one you use, you're going to come up with different answers.
Yeah, we will get back to that question later on, because I also have a question on speciation. But another question about what you said about sperm competition. I mean, that also occurs among the sperm of different males of the same species, correct? If they just inseminate the same female.
Yes, yes. Yes, so there's a fascinating experiment with fruit flies with that, in which they set males to evolve without the gene flow from females. So they use some genetic tricks that only the father's gene would stay in that compartment, and the female compartment would actually never see genes from that compartment. And those males evolved to be exceptional sperm competition winners. So when they compete with males from the other population, which have been co-evolving with females, they always win. And that is to the detriment of the females. The females actually have a shorter lifespan after mating with these males. So there is a trade-off. The trade-off is the longevity of the female. But if you think in terms of the selection that went on in that compartment where only males were being selected for, right? It doesn't matter, because in a promiscuous system, the male will get eggs from that particular female early on, and how long more she lives is not going to affect how his genes do. So you can have these, it's called antagonistic sexual selection, in which what is good for one particular sex is not necessarily good for the other sex. And you have, you can even think of in terms of sexual selection, right? And you have like some, let's say, a fanciful tail that females find really attractive, and they mate with that individual, and perhaps they mate with that individual in a part of the season where they shouldn't be reproducing, let's say because winter is closed. Her offspring may survive, but because she laid these eggs early on the season or late in the season, she may not survive. So selection actually acted against the female from mating with this particular male, even though in favor of the male for having a few more offspring because of his fanciful tail. So in this case, a case of antagonistic sexual selection, in which the female should then evolve resistance to that particular trait. And then you see these amazing things where, like the swordfish, the females with a sword, the species with a sword, the females don't care much about the sword. But if you artificially add a sword to a species that does not have a sword, then the females go crazy for it. So the species that don't have a sword are very attracted to swords. The species with swords, females don't care about it anymore. So it seems like one of those cases that females eventually evolved resistance to a trait in which at first they were attracted to. Yeah, there is some very funny spider examples too, with like the females like hairy spiders, but in species where they're really hairy, they don't care about hairiness. And you can actually play videos to the female, and they respond to videos, and you can modify the males to have extra hairy or no hair. Watch how the females respond, which is very funny.
Yeah, it's very funny. What about the case that I mentioned of interspecies gene transfer via infection, for example? I mean, why is it that in some cases the genes really become part of the genome of the germline cells and other times they don't? I mean, how do they get there exactly?
So I think that it's chance, right? So if you think of infections, they happen so often. Like you think like, how many times during a pregnancy a mother is going to get infected, and the infection potentially infects the developing embryo, or even like in animals that lay eggs, like the case of the aphids. Oftentimes these eggs get parasitized by fungus and whatnot during development. If it doesn't kill the embryo, then this embryo has the capacity of receiving that, right? So I think that what we're looking at is an event that is so incredibly common. Infections are so incredibly common that eventually it sticks, right? It's like if it happens a million times, once it's going to stick. And as long as it's not detrimental to the species, then it has a chance of acting like a new mutation, either going to fixation, and at some point in the future everybody has it, or to loss, and at some point nobody has it. And yeah.
So let's talk about another topic. What is modular expression of genes?
Right. So that's when, if you have like a particular phenotype that always tends to go together. So for example, a nice example is these burrowing mice, and they bury long tunnels with an exit, a escape exit at the end, or short tunnels without a escape exit. So it seems like the two things, the exit, the presence or absence of the exit and the tunnel length, they go together. They must be regulated by the same kinds of genes. They should be one and the same. When you create hybrids, these are two different species. You can create hybrids and actually map the presence and exit of that escape tunnel and the length of the tunnel onto the genome, and you find that they are regulated by completely different and non-interacting sets of genes. So that if you create hybrids, you can have hybrids that build a short tunnel with escape or a long tunnel without escape. So this is the modularity. The genes act as independent blocks, adding on behaviors to existing behavioral sets without interfering with other things that were there. So that's the modularity of some interactions. Of course, there are genes that interact with each other, and the presence of one modifies the effects of another one. But in many cases, they are modular.
Mm-hmm. So it's obviously not only about the genes a particular individual or a species has, but also, and perhaps even more important, what genes and how they get expressed.
Mm-hmm. How they interact, yeah, exactly. So of course, the genomic background matters, right? So sometimes a particular allele that causes a particular phenotype in a given population, when you insert that allele in another genomic background, they do something else, or they are detrimental, etc.
Mm-hmm. So, and I mean, can this also lead to speciation or not? I mean, in the sense that perhaps two different individuals have the same set of genes, but because in one of them particular modules are expressed and in the other not, I mean, could these have something to do with speciation or not?
Yes, if they are related to any kind of creating barriers to gene exchange, right?
Oh, okay.
So if they are, for example, acting on when the individual matures to reproduce, what time of the year, and that automatically creates a barrier, then yes, they could.
Yeah. So, okay, so we've already touched on the concept of species. We're going to get into speciation again later on, but another very disputed concept in evolutionary biology. What about adaptations? I mean, what is the best way to understand how adaptations evolve? And I mean, is it better to try to understand it at the individual level or the population level?
So we, selection will obviously act at the level of individual, but the effects are always observed at the level of populations. So if you're talking about local adaptations, right, you want to find populations that share a particular local adaptations. So a fascinating example in humans is this fishing population in the Asian Ocean, where males will hunt for fish in the ocean and bring it back to their kids, and they can spend an amazing 11 to 13 minutes underwater without breathing. I mean, this is just like, after a minute, I'm panicking. Like 11 minutes is a lot. They mapped this gene, right? They were able to map this gene to a single nucleotide substitution. It's a local adaptation to this particular mode of life, where individuals, because they depend on the sea, that could spend more time underwater, were selected in favor. They had more surviving offspring. Their wives could reproduce more often. And eventually, this particular allele went to very high frequencies in this local population. So this would be a local adaptation to a particular lifestyle. Mm-hmm. You have oftentimes a particular adaptation might be fixed in a given species. So everybody has it. So in the case of my crickets, the ones that live by the beach have a very long ovipositor. So it looks like a tail that the female uses to lay eggs deep in the sand. Because it's sand and it's going to be blown by the wind, it needs to be deep. Now you think of the same, it's about this big, right? You think of the same length in a clay soil. It's not going to go because the female will likely break as she's trying to get through rocks and whatnot. And there is no need. If the eggs are that deep, they're probably going to have a hard time coming out in the spring. So different adaptations, the clay soil population has tiny ovipositors compared to very long ovipositors from the beach population. So another adaptation, but in this case, there is no variation within the species. It's more like the adaptation goes for the entire range of that particular species. So there's not as much variation in space within a given species, but a lot of variation between species.
Mm-hmm. But I mean, for something to be considered an adaptation, it has to spread, even if it originates in a single individual, it has to spread through the population, right? Because otherwise it wouldn't be an adaptation, because an adaptation also has to at least increase reproductive ability fitness. Mm-hmm, mm-hmm.
Yeah, but it doesn't need to necessarily spread to the entire species, right? So, yeah, the species. So in the case of this fishing population in humans, it's very limited. And I think that there are trade-offs. So the individuals that are able to spend 11 minutes have a much larger spleen, and they store like oxygenated blood in the spleen. But you know that you can rupture spleens. It's actually a medical risk to have a very large spleen. So it might be there might be a trade-off between the diving ability versus, I don't know, like I know a person that ruptured her spleen by riding a horse. So, you know, maybe there is some kind of trade-offs of that sort. So it need not spread to the whole species if the adaptation is local. So humans also have adaptation to live in high altitudes. That has not spread to the whole species, right? Mm-hmm. Whereas you, of course, you do have adaptations that the entire species share it because it was advantageous everywhere they exist. So as long as it's advantageous everywhere, then it should spread to the whole species.
Mm-hmm. Yeah, I mean, evolution by natural selection never operates at the level of the species, right? I mean, that's an ancient idea, perhaps from the late 19th century, early 20th century, but it's already been discredited, right? Right. Mm-hmm. We have to look at the population level. Right. Mm-hmm. And the selection is acting at the individual level. At the individual level. We see it at the level of population, acts at the level of individual. Okay, very well. So, how do evolutionary biologists and evolutionary geneticists go about identifying the genes associated with a particular adaptation? So there are several methods, right? A method that is used less and less often is to find candidate genes. So let's say, you're trying to find adaptations to life on caves, and you have albino fish that are also blind. You know the genes related to melanin production. You could go directly to these genes, sequence it, and see if these cave populations are any different from the surface populations. And then you can do a series of selection tests to see if that went under selection or recent selection. The problem with that is that this example I picked is an easy one, the melanin production, everyone knows. The problem is that most adaptations, we have no clue, right? What allows a human to stay underwater for 11 minutes? We don't have any clue. Can we find a gene related to that adaptation in whales? We wouldn't even know where to start. We don't know. So this method has yielded some good results, but has been largely unsuccessful in finding genes, and has been replaced by more genomic-wide methods, right? So if you have two species and you want to find out, for example, what are the genes that cause mice to build an escape on their tunnels, and this is not learned, they are born either doing the escape or not doing the escape, which is very interesting. You wonder what goes in their heads, right? I must do escape if you have the gene, or otherwise not. You can create families, because these behaviors are in different species. You can create hybrid families, hybridize them for 2 generations. And now the offspring have a range of behaviors. Some build the escape tunnels, some do not. You sequence the whole genome and you find associations between particular positions in the genome and the presence of escape tunnel.
So if you find that every single individual out of, let's say, 300 individuals that did build a tunnel also have an A at this particular genomic position rather than a T, then there will be a high association between the presence of an A and the escape tunnel. Let's say that all of the ones that did not make escape tunnel have a T or a T and an A. So that would be a very high evidence that that position in the genome is highly associated and might be the gene behind digging exit escapes. So in this case, you're making families in the lab, and families in the lab are essential if there is no variation of the trait within a population. So if all the individuals of one species have, let's say, a long ovipositor and all the others have a short ovipositor, the only way that you're going to get variation is if you mate them together, create hybrids, and then examine the hybrids. If there is variation inside populations, like for height, for diving ability, how long can you stay underwater, all these things, then you can actually use unrelated individuals that you didn't produce a family from the population and do the same thing. So you measure a particular trait and you associate with the genetic variation in that population. And that's what they're doing for humans, right? Every day there's a new paper on GWAS, genomic-wise association study. So it's exactly that. You just get a population that varies in food preferences. That's the newest one, right? And you map what is associated with food preferences, and you find the differences. They're significantly associated. You would never find that by chance. So chances are that those loci are playing a role in the particular behavior.
Mm-hmm. Yeah, they do that a lot in behavior genetics, for example, and studies done with twins and stuff like that. Right. Mm-hmm. Yeah, so now that we have very cheap ways of sequencing whole genomes, especially for humans and model organisms, it's very easy. Like for my crickets, it's much harder because we don't have those tools. So we can use like shortcuts like DD-RADS and other things that just look at thousands of positions, but not the entire genome, and that gets you somewhere. Yeah, and I guess that another complication here when it comes to identifying the genes associated with a particular adaptation, a particular phenotypic trait is polygyny. I mean, many traits are polygenic. There are multiple genes give rise to them, and then we also have pleiotropy, that is, the same genes participate in giving rise to multiple traits, right? Yeah,
which is very common even between human diseases and in particular behavioral or diseases conditions. You have a lot of overlap, right? So you say genetic correlation. So you have autism correlated with schizophrenia correlated with something else. So you have same genes that are participating in very different phenotypic conditions, yes.
Yeah. And perhaps some of them, I mean, perhaps some of the people that are predisposed to different conditions, it's because the same aspect of physiology, for example, that gets affected, affects the brain in those two different ways, right?
Right.
Yeah. So let's get back to speciation. I mean, how can we know that speciation is occurring?
So I think, as I mentioned before, you have a continuous, right? So you have no differentiation between populations at all. You can have a species in which you look at different locations in the landscape and you see that the populations have different gene frequencies or allele frequencies. And they're not different species, they're fully fertile. You know, it's the case of human populations, right? There's differences around the world. We're all fully fertile, we're not different species. So that would be a genetic structure present, but not speciation. If you continue to build in genetic structure, and genetic structure requires some kind of reduction in gene flow. So the genes have the gene is not flowing as much between species as it is within a given location, right? Usually because of geographic barriers, mountain ranges, rivers, etc. The consequences is that over time, if that barrier doesn't disappear, the populations will become ever more differentiated. And it's very hard to say when speciation happens. Obviously, at the point that barriers to gene exchange are present, and there are many of them, then you can say, yes, the speciation has happened, but exactly telling now it happened, it's difficult because it's a continuous process, right? And these different species concepts will apply. So one of the species concepts, for example, is that the species fall in unique genealogies, so unique evolutionary trajectories. So this can happen much earlier than the development of reproductive incompatibility, and much earlier than the development of hybrid inviability or post-zygotic isolation. So there is a continuous, and depending on which species concept you're using, you're gonna break it differently.
Yeah, that's the issue, right? Because even the concept of species is still debated.
Well, there are dozens of them, so it depends on.
Yeah, and I mean, it's interesting because we don't really need to have a concrete definition of species, like we didn't need to have one for gene before we knew what a gene was, and people were already we're already discovering things about how genetics worked, right? Even without knowing what a gene was.
Right.
Yeah. So what is phylogeography?
So phylogeography would be the study of genetics over space. So you can, interesting phylogeographic studies will look, for example, at various populations over, let's say, North America landscape, or European landscapes, or anywhere really, South America. And oftentimes what happens is that you see concurrent patterns. So in North America, for example, you will see that the South, and the same goes for Europe and Asia, is richer, has more genetic diversity than the North. And if you understand the evolutionary pattern of these species, you understand that the North was glaciated, and therefore the northern populations did not exist until more or less recently. And they had to migrate after the ice goes away. And these migration patterns usually happen by just a few individuals of the existing large populations moving a little further, and then moving a little further, and then moving a little further. So the further away you go, the less genetic diversity there will be. And again, in the human populations, it's very clear, right? As you move from Asia into the Americas, you have a huge decrease in genetic variation. So the American population, the original American Native Americans were colonized through the Beringia, and as they went down, they lost a lot of genetic variation because few individuals moving down. You can't have all the representation that existed in Asia before. Right?
Yeah. Does that have anything to do with latitude, and perhaps certain latitudes leading to, for some reason, more diversity, more species diversity or something like that?
Yeah, so obviously, if you look at the Amazon rainforest, right, you see a lot of species diversity. So tons of species. It has been a continuous environment that has been able to support life for much longer than, let's say, Canada boreal rainforest, because it didn't have the same glacial problems. And it did have, so one thing that is important is the allopatric feature, right? So if you have extrinsic barriers and you can isolate populations for a while, then differentiation builds up in those isolated populations. Perhaps to the point of first barriers appearing, right? The genomic divergence that is so big that when they get back together, they cannot produce fertile offspring, and they're good species now. So something like that may have happened in these areas with huge numbers of species. They have been continuous for a long time, and in the past, they may have been broken up because of more arid climates or something like that. And now that the forest expanded, the species are able to be together again, but no longer able to produce offspring.
Does it have anything to do with it being tropical areas or something like that? I mean, with basically the environmental conditions that lead to more diversity in terms of species?
There is debate. So it's interesting that if you're looking like the far boreal forest for some groups like birds, nowadays they are speciating even faster than animals in the Amazon. And that could be because it's an environment that recently opened, a few species went there, and there is still way more purine species. But if you look at how old the species are, they are relatively recent in the north and older species in the south, and it could have something to do with like, the problem is that, the latitude has been all over the world. We don't have replicates in which the north latitudes have been continuous, right? So they all have the same pattern. So it's hard to actually test that.
Yeah, I understand. Okay, so one last topic, phylogeography. What is landscape genetics?
So landscape genetics is very similar to phylogeography, but usually in contiguous populations in much smaller space, contiguous collecting. So in the study I did with artic plants, for example, we collected in the entire range of this. It's a disjunct population in Upper Lake Michigan and the Isle Royale National Park. And we virtually collected every single individual within that landscape. On top of that, you collect ecological variables. So where are water differences, soil differences, presence of rivers, like very local kind of thing. And you don't have to classify, the collection point of every individual you did. So you don't have to call, oh, this is a population, this is a population. You know the location of the individual. So that's what you're analyzing, is the known location. The known location and the known landscape features, and you're trying to find evidence of migration breaks, for example. So for the Arctic plants, it's easy because there are islands. So the islands obviously have a lot more migration within them than between them. But in addition, you find that currents in the lake will play a big role in migration between islands. So some islands have a lot of migration, some islands do not because of the prevailing currents. So it's these kind of things, like very local phylogeographic studies. So for phylogeographic studies, I once did a study with bark beetles in the entire extension of Canada, everywhere. And for that, we didn't know the exact location of each individual. We had, one county, the other county, the other county. We called those populations, right? And where we found the break was between different hosts, pine tree hosts, spruce hosts. So depending on the host, you would find breaks in the genetic structure, which would suggest that the host tree creates some isolation. So there might be local adaptation for particular host species.
So, I mean, we can say that perhaps geography and the kinds of landscapes, different individuals, different species or populations find themselves in affect or influence gene flow, right? Perhaps it's easier in certain landscapes for them to have more gene flow than others. Right?
Sure. Yeah. So depending on the landscape features, there will be huge differences, and sometimes it's as small as a different soil type, right? So some landscapes have very clear partitions of soils. You go from a very calcium-rich soil to one that is not as rich, and that alone can create huge barriers for some plants, some creatures, some pollinators. On top of that, it's interesting that pollinators actually learn to feed on particular plant species. So in a given location where that species is common, most pollinators maybe use it as the main source, whereas in another one where it's rarer, the pollinators may not use it, and that alone could influence gene flow and so on.
Yeah, and perhaps there are certain instances where the features of the landscape is at least one of the factors that gives rise to speciation, right? Because, for example, if individuals from a certain population migrate to a different area, and then for some reason, because the landscape changes, they basically diverge into two different populations. I mean, that can lead to speciation, correct?
Sure. Over time, it can definitely lead to speciation. And I think it's fascinating when you find, I actually had that in my final exam for my students, right? You'll find the grizzly bear population structure is very structured. So in British Columbia, it has like 3 populations that are not exchanging many genes. So if you look at the genome as a whole, they form 3 very distinct populations, indicating limited gene flow. But then when you look at the Native American languages, it follows almost exactly the distribution of the bears. So indicating that the barriers that acted on bears and prevented them from mating with other bears also acted on humans and prevented them from exchanging language with other humans, right? So so the same barriers can actually act over different species, which is very cool.
Yeah, this is very interesting because, I mean, if we get into anthropology, we can also link genetics to linguistics to culture, for example, right, and understand, the timeline of how 2 different groups of people split and gave rise to 2 different languages or cultures or whatever.
Mm-hmm. Yeah, no, it's fascinating.
Yeah, so hopefully I also passed the exam today. Okay, so Dr. Maroja, before we go, would you like to mention where people can find you and your work on the internet?
Sure. Yeah, I work at Williams College. My email is lsm1@williams.edu. Yeah, if you have any questions, anything, email me.
Okay, great. So I will be leaving some links in the description box of the interview, and thank you so much for taking the time to come on the show. It's been a real pleasure to talk to you.
Yeah, no, thank you so much.
Hi guys, thank you for watching this interview until the end. If you like what I'm doing, please share it, leave a like, hit the subscription button, all of those things you already know. And please consider supporting the show either on PayPal or Patreon. All of the links will be in the description box of the interview, starting at $1 per month, so it would be a great help. This show is brought to you by Enlights, Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters: Karen Litske and Blanchett, Pererga Larson, Lau Guerrero, Francis Ford, Hans Frederick Sunder, Ricardo Vladimiro, Greg Healy, Adam Castle, Olaf Alex, Jonathan Wiesel, Jacob Klinkby, Matthew Whittingbird, Arno Wolf, Tim Hollis, Eric Alenia, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Vosbo, Weingard, Rebecca Newberger Goldstein, Dan Demetrio, Robert Windager, Rui Narcio, Arthur Coe, Zup, Marco Neves, Colin Holbrook, Susan Pinker, Bernardo Seixas, Pablo Santurbano, Simon Colombo, Jorge Espinha, Phil Kavanagh, Mark Blythe, Roberto Inguanzo, Michael Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Ugni, Alexander Dunbauer, Fergal Cusson, Evan Bodrenko, Al Herzog, Don Ross, Jonathan Librant, Oslam Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, David Sloan Wilson, Yacila Dez Araújo, Eden Solon, Romain Roach, Dmitry Grigoriev, Diego Londoño Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Hellman, João Linhares, Lida Cosmidis, Sam Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa, Jules Price, Edward Hall, Edin Bronner, Franca Bortolotti, Gabriel Pons Cortez, Ursula Litske, Denise Cook, Scott, Zachary Fish, Tim Duffy and Trader in NYC. My producers Izar Webb, Jim Frank, Lucas Stafiniak, Ian Gilligan, Luis Caetano, Tom Vanagdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardus France, Thomas Trumbull, and Nuno Welder. And my executive producers Michel Rugieski, Rosie, James Pratt, Matthew Lavender, Sergio Codreanu, and Bogdan Kanivets. Thank you for all.